Теперь, когда все технические проблемы решены, мы можем начать наше заседание. Значит, заседание диссертационного
Session of the Dissertation for the defense of the thesis by Gurevich Alexandrovich for the presented for the degree of candidate of Physico-Mathematical Sciences, specialty 030101, Mathematical Biology and Bioinformatics. Let me declare the session open. According to the order number 9559, issued on 3rd of October 2018 at the St. Petersburg State University, me, Podgorna Olga Igorevna, Doctor of Biological Sciences, Professor of the Department of Cytology and Histology of St. Petersburg State University, was appointed the chairman of this dissertation board. Let me introduce my fellow board members. Mikhail Sergeyevich Gelfand, Doctor of Biological Sciences, Kharkiv Institute for Information Transmission Problems, Russian Academy of Sciences. Online, O'Brien Stephen James, Doctor of Biological Sciences, St. Petersburg State University.
Steve, will you please stop drinking tea? Steve, will you please say hi?
Hi, how are you? I hear you perfect.
Thank you.
Felipe Adam Michael, Doctor of Biological Sciences, National Human Genome Research Institute, USA.
Yes, hello.
Okay.
Monix Henrik Medema, Doctor of Biological Sciences, Wageningen University, the Netherlands.
Please say hi. Okay, thank you.
According to the federal law number 127 of the 23rd of August 1996 on science and the state scientific and technical policy, St. Petersburg State University has the right to independently award academic degrees. The corresponding order on the procedure for awarding academic degrees at the St. Petersburg State University, on the basis of which we are holding this session of the board, was issued on the 1st of September 2016. Order number is 6821/1, hereinafter referred to as the order. In accordance with this order, the session of the dissertation board is duly constituted if at least 2/3 of the approved members of the board are participating, but not less than 4 persons. Our dissertation board consists of 5 members. All of them are present, including 3 board members who are working online in remote interactive mode. We checked the audio-visual contact has been established. Thus, we had the necessary quorum. Let me set forth the following procedure of today's session. The total duration of the session shall not exceed 2 hours. We shall start with Chairman's summary report on the main content of the documents submitted by the candidate and their compliance with the established requirements. That will be followed by answers to possible questions, a brief presentation by the candidate for the degree outlining the key points of his study, no more than 15 minutes. Questions to the candidate strictly related to his presentation, no more than 2 minutes for each question. Responses of the candidate. A reading of an external review by the chairman. The review was posted on the SPB-SU website. Speeches of all the members of the dissertation board who will present their reviews, opinions, questions and suggestions for the candidate in turn. No more than 10 minutes for each speaker.
will be the first. Выступление, так,
the chairman's presentation of his review, the candidate's answers to the questions and remarks made by the members of the dissertation board, no more than 15 minutes. An open discussion. The attendees may briefly outline their attitudes and ask specific questions and suggestions for the candidate strictly on the topic of the study, no more than 5 minutes for each speaker. To do that, each speaker is kindly requested to put his name on the registration sheet and fully introduce himself before making a speech. When the procedure is clearly outlined, no one gets any questions. Don't be scared. Feel free to ask questions, please. Because if we get no questions, everyone gets an impression there's nothing to discuss here, and that's a very interesting paper. Final answers to the candidate to the questions asked no more than 5 minutes, a reading of the review by the academic advisor, a 5-minute discussion before the open voting procedure on awarding or not awarding the St. Petersburg State University academic degree to the candidate. For the time of this discussion, the board members, the broadcast shall be suspended. The individual voting, the chairman of the dissertation board shall count the voices and the results shall be recorded in the minutes of the meeting. Decision on awarding or not awarding the SPBSU academic degree to the applicant and the final word of the applicant, no more than 2 minutes. Before starting the official procedure, let me remind you to switch off your mobile phones. Thank you for your understanding. Let me also inform you that our meeting shall be recorded and broadcast online at the SPBSU website, and the speeches shall be simultaneously translated from Russian into English and from English into Russian as appropriate. Let's start with the first point, the chairman's summary. The dissertation of Alexey Gurevich, Computational Methods for Analysis of Error-Prone Metabologenomic Data, submitted for procuring the degree of the candidate of Physico-Mathematical Sciences, Specialty 030109, Mathematical Biology and Bioinformatics, was accepted for defense by the order issued on the 3rd of October 2018 by the SPBSU Academic Secretary. Order number is 9560/1. The dissertation board was appointed by the order 9559/1 issued on October 3, 2018 by the SPBSU Academic Secretary, and I have already introduced the members to you, so you know them now. The candidate has submitted the following documents to the Academic Secretary. An application to the Rector of SPBSU, Professor Kropotchev, to accept the thesis for review and defense, submitted on the 1st of June 2018. A list of 11 publications describing the main academic results of the thesis. All the 13 papers describing the main academic are published by the bodies including the International Database and Citation System, the Master's Diploma ON 12589, issued by the St. Petersburg Academic University of the Russian Academy of Sciences on the 8th of June 2012, where Alexey Gurevich was awarded the Master of Science degree in Applied Mathematics and Physics. A certificate on passing the candidate's exam, Specialty 030109, issued by the Institute of Information Transmission Technology of the Russian Academy of Sciences on the 9th of November 2017. a review by the academic advisor, copies of the thesis in English and in Russian as manuscript and in the electronic form. The thesis was submitted in both two copies, in Russian and English. The thesis has been formatted according to the requirements of the National State Standard R7011. The thesis has been printed on a multiplier with an indication of the output data in accordance with the National State Standard R7.0.4. All the listed documents comply with Article 12, Section 3, Thesis Presentation and Defense of the procedure for awarding the academic degrees at the St. Petersburg State University. Appendix 1 to the order. If anyone has any questions related to the documents listed above, let's move to the next point, the applicant's presentation. That will take 15 minutes.
To present you key points of my work. The motivation behind this work is the search for natural products which have a great potential to become new medicines. It's known that short molecules produced during metabolism in plants, bacteria and fungi are common examples of antibiotics, anti-tumor agents, and other clinically important compounds. But their discovery is a complex problem, and modern biotechnologies can help them a lot. For example, sequencing technologies allows one to sequence genome of natural product producing organisms, but these results of the sequencing are short fragments of DNA, and it's difficult to find genes encoding natural products. So computer scientists help biologists by developing genome assembly software that allow to combine short fragments into larger fragments of DNA. Further, genome assembly evaluation methods can be applied to select the best assembly. And finally, genome mining approaches are used to predict gene clusters that encode novel natural products. In practice, usually when we work with bacteria, it's difficult to cultivate them in the lab, so sometimes researchers work with so-called meta-genomes, where a huge set of different bacteria are analyzed together, and in this case we talk about meta-genome and meta-genome assembly as a DNA corresponding to all these samples. At the same time, modern mass spectrometry techniques allow to analyze metabolome. Sorry?
Nothing. That microphone's thrown away. Use another one. Sorry.
Okay.
The audio is good for me.
Okay. So mass spectrometry techniques allow to analyze metabolome of this organism, but the problem of identifying natural products from mass spectrometry data is also challenging. So computer scientists need to help this direction too. And one of possible options is dereplication, which literally means removing of replicas, of duplicates, where we compare mass spectra with already discovered compounds and significantly reduce the complexity of input data. After this, one may try to predict the structure of novel drugs based on mass spectra, but it's still a challenging problem. So another option is variable dereplication, where we search not for exactly previously discovered compounds, but of their variants which occurred due to mutations or modifications. If you look at the whole figure, we can see that there are two routes to the same goal. So if we combine together them, we can significantly help natural product researchers. But creating of this pipeline is difficult because almost all these errors represent a computational problem, and very difficult problem. Some of them have already mature software solutions like genome assembly problem, but some of them are still open. So in my dissertation, I address problems from both halves of the pipeline, and I will tell about them in more details. And let's start from genomic part of the pipeline. The first considered problem is genome assembly evaluation problem, and prerequisites for this problem is the presence of many sequencing technologies with different output, and of course many genomes to sequence. So there are dozens of genome assembly algorithms which produce different assemblies, and we need to compare them and to evaluate and decide which one is better for each particular case. But existing genome assembly comparison and evaluation tools have a lot of limitations, so there is a need for universal and easy to use tool, and such tool was developed, and in my work I present Quast and how it solves all these limitations. For example, it works with any organism from all kingdoms of life. It can work with and without finished reference genome, so you can apply Quast to just sequenced organisms. Next, it allows to assess quality from different points of view and compute a lot of metrics and draw important plots. And finally, there are a lot of ways to install and use Quast, and even researchers without much computational background can use it easily. The next problem is meta-genome assembly evaluation, and it's known that just a few bacteria can be cultivated in the laboratory, so there is a need to assess and analyze genetic material taken directly from environment, and in this case each sample contains a lot of thousands, hundreds species with different abundance, and the same is true when we assemble this data. So various assembly algorithms again exist, and they demand a new benchmark procedure, but the evaluation of meta-genome assemblies is even more complex than the evaluation of single genome assembly, and to solve this problem, MetaQuast software was developed, and it handles all common challenges, like it deals with unknown species content by detecting the species content of the sample based on 16S RNA. It also automatically downloads reference sequences from large biological databases. It can work with unlimited number of reference genome, and regular Quast visualization was significantly redesigned to support reports for multiple genomes. And finally, it can detect errors of assembly software when they incorrectly join together fragments from different, albeit closely related organisms, and also can separate true assembly errors from false positive caused by structural variants. That's all about genomic part of the pipeline, and let's move to metabolomic. And first considered problem was natural product dereplication problem, and in particular, in my work, I focused only on peptidic natural products, which are important class of natural products and consist of non-ribosomal peptides and ribosomal peptides with some post-translational modifications. This type of compounds include many important medicines, including antibiotics, starting from the very first penicillin and ending with the currently last teixobactin, which was discovered just a few years ago. And at some point, discovery of novel PNPs became very slow because of high rate of rediscovery of previously found PNPs. So early performed dereplication, removing of these previous copies, minimizes a lot of resources, saves a lot of resources. At the same time, PNPs significantly differ from regular linear peptides. In particular, they have non-linear structure, mostly cyclic or branch cyclic, and often contain non-standard amino acids or modification of standard amino acids, and this prevents use of standard proteomics software for this problem, because proteomics tools are focused on linear peptides consisting of just 20 standard amino acids. So the new program was needed, and in my work, the replicator was developed. It starts from an experimental spectrum and a database of known PNPs. It scores spectrum against all PNPs from the database with matching mass and score this so-called PNP spectrum matches, or PSMs. Further, it computes statistical significance of each score and report identifications which are above some threshold on the significance. This pipeline already looks complicated, but it's only half of the pipeline. Actually, to estimate false discovery rate of this tool, we also generate decoy database, which consists of randomized version of target peptides, and we consider that each hit in this database is false positive because compounds in this database are artificial. We perform the same steps with this database and finally estimate FDR as the ratio of number of decoy hits to number of target hits. Final problem is variable PNP dereplication, and it is important because it's observed that PNPs are extremely modified and mutated, and most PNPs form large, sometimes very large families of related peptides, while databases contain only few, the most abundant representative of these peptides, which were easier to identify. So, at the same time, rare PNP variants may be more clinically effective, as shown by recent studies. So, variable dereplication is crucial for PNP discovery, but this is a complex computational problem because the set of modifications and mutations is large and not known in advance, and also spectral dataset may not contain any known PNP, only its variants, so we don't know where to start our search and need to consider all PNPs from the entire database. So the search space for this problem is much higher than for standard dereplication. To solve it, VarQuest software was developed. Similar to original tool dereplicator, it starts from spectrum and PNP database, but the first step is preprocessing of the database into a small compact form, which allows to quickly search spectrum against the entire database and find the most promising candidates, which are further rigorously scored, and we apply all possible positions for modification, score them against the spectrum, compute statistical significance, and finally identify PNP variant. That's all about the main problems and their solution, and I would like to show some slides about approbation of this work. All 4 tools were presented at leading international conferences in computational biology and bioinformatics, and also all 4 were published in international journals, including bioinformatics, Nature Chemical Biology, and Nature Microbiology, and got a lot of citations, especially the first one. Also, Quast package, which includes both Quast and MetaQuast, is highly popular and used in 1,000 laboratories right now from various countries, while Dereplicator and VarQuest were added to GNPS core in silico analysis tools, where GNPS is an international platform started a few years ago, and which combined researchers of natural products from all over the world. And now it contains more than 1,000 different mass spectrometric data sets, and we were able to benchmark both our tools on more than 100 million mass spectra and identify a lot of known and compounds and their new PNP variants. Finally, as a conclusion, I would like to show you this slide one more time and say that my future work is focused on the combination of both parts of the pipeline and creating a full pipeline that will transform PNP discovery into highest output technology. Finally, I would like to thank all my coworkers, both from Algorithmic Biology Lab at St. Petersburg Academic University, where this work was started, and St. Petersburg State University, where this work was finished, and also my international collaborators, and of course my scientific supervisor, Professor Pavel Pesner. And thank you all for your kind attention, and please question me. Thank you.
Questions, please. Okay, let's move to the reading of reviews. We received an external review by Lagunin Alexey Alexandrovich. The Russian National Research Medical University, named after Pirogov. The review was duly posted on the SPBSU official website as required by the Article 19 of the Procedure 40 Academic Degrees at the St. Petersburg State University, Appendix 1. I will summarize the review briefly. Lagunin says that the author has very well described the current state of this relevant problem. In his thesis, he found solutions to the tasks he set forth, and in both cases, he suggested he is offering solutions and new software products. The author emphasizes that the new software was so high quality that they have been cited in the world by the world popular publications, and they're now widely used in the world. The author says that Alexey Gurevich's work has been verified by a vast amount of data, and the results appear valid and trustworthy. His thesis is well written and well organized. The theoretical papers done by the author independently at a high professional level. He would like to make two small comments. Table 1, page 34, does not contain the results illustrating NA50 as compared with NGA50. That would demonstrate the need for entering the new parameter NGA50. Tables 2 and 4 should show the results of the work of other software used for genome assembly assessment, genome and meta-genome assessment, which would enable us to evaluate efficiency of quast and metaquast as compared to other well-known software. The author says that these minor comments are not criticism and cannot affect and do not affect the overall high evaluation of the thesis by Alexey Gurevich, which meets the main requirements set forth on the order on the procedure of awarding academic degrees at the St. Petersburg State University. Of the candidate of Physical Mathematical Sciences, specialty 030109, Mathematical Biology and Bioinformatics. In that case,
Steve, I think they're waiting for your review.
Oh, I'm sorry. Yeah, I heard. You want me to go first? Okay. Are you kidding me? Lexi, where are you?
Yes, please start your review.
Yeah, why don't you just stand there so I can look at you while I'm speaking, if you don't mind? Okay?
Okay.
All right. First of all, I read this thesis and enjoyed it, even though it's a technical triumph. I did understand what you're saying the thesis did, and I agree with the previous comments. Alexey moved again. Alexey, could you stand at the podium?
Sorry?
Yeah, just wait there. Are you listening?
Yeah.
Can you hear me okay?
Yeah, thank you.
Okay, as I said, I think the thesis was pretty well done and understandable for lots of people, and I congratulate you for that. I recognize there were technical details of chapter 1 with the introduction of the class algorithm as well as the assembly. And I for the whole thesis because I just did that, but I will give you a question because I was supposed to 2 minutes. And the first question is, what actually the compound identified in the metabolome search dereplicator? What were they? What was the actual outcome? The top 10 why were they described as important in your work?
Please repeat the main part of the question because there was some technical issues.
Steve, will you please repeat the exact question because of technical problems?
Sure. My questions are, most of the thesis is methodology. Most of the thesis is technical. The programs released are useful. You apply them and you specify in your presentations future application. So tell us, what did you discover? Compounds? Why are they important? What are they important for? You understand the question?
Yeah. Sorry. If I understand the question correctly, you want to hear from me what is the importance of these tools? So why?
No, I want to know what compounds you discovered and why they're important.
Yeah, I see. In VarQuest paper, we discovered a lot of PNP variants, new variants, but our results of our work is a kind of intermediate step between raw data and biologists and researchers who will explore their biological activity of these compounds. So I cannot say right now what is the biological importance of the discovered compounds. I just so our tool was able to identify a lot of structures, some of them experimentally confirmed, but we don't perform any clinical testing or something like this. So it's out of scope of this work. This is more computational work.
Okay.
Are you satisfied?
If you take 10 million compounds in mass spec and you come up with a listing of 10, 100,000, why are the top 10 more interesting than the other 10 million? You understand the question?
So we search against a database of already proved biologically active compounds which represent drugs and antibiotics, anti-tumor agents. So we searched against an interesting target database. So when we find something similar, it's a high chance that what we found is also have some important features. So that's because we search against already important compounds, including clinically important.
Clinically important for what?
For different ways. For example, antibiotics, which are active against Streptococcus aureus, so Escherichia coli, and so on. For example, these ones. or against some plant pathogens and so on.
Natural products has been around for a long time, and it has been promising to discover many things. Some things have been discovered, like tactile, but many of the things discovered have really been any better than the ones that have been thought about already. Tests in laboratory activity against cancer cells, HIV, or bacterial growth. So how many real successes out of computation, bioinformatics, metabolomics, like you're proposing? Are there any real examples today, or are we still waiting?
So I think the real examples, so we show some small examples which we found in our search, which was published just in this year, and I hope that more examples we will have later when some more biologically background person will try to analyze our results. So we just provide a list and structures, and the next guys in the pipeline should analyze them. So we simplify their work because they don't need to analyze all spectra, but need to analyze only a few of them.
Okay. Alexey, because this is your PhD exam, I promise you, you will remember everything your examiners say from top. Because your work. So I have nothing to say based upon your answers to my question. Prepare in the future a listing of the real application and prepare a list of discoveries you find that you think are important and why. And third, prepare a listing of peptides, PMPs, that have been used by this algorithm with 100,000 downloads that are taking place by other people, so that funders and the viewers of your program will, even if they don't understand the technology of the algorithm development, the improvement, fine-tuning, application, you know what the target is. That's a comment, not a question. I have one final question, which is,
so I have one example in additional slides like this one. So in our work we present an example of a variant of macetolide A. So macetolide A was a known compound which has some activity against a specific pathogen of plants, and we found a new variant of it, and computationally. And after that I searched in scientific literature and find that this variant was just discovered a few years ago, so our database was older than this, and this new variant was have also activity against more cell-borne plant pathogens, and also have an additional function of colonization of sugar beetroots. So this new variant is more interesting, has more features, and it was discovered just in a few minutes by computational search of the data. And we have a little bit more examples in the paper. Yeah.
Good. Have you and Pevsner and all your colleagues working on this project together begun to start thinking about the next step, which is laboratory replication or validation of biological activity of any of these compounds? And if so, tell me a little bit about that.
So we don't plan to do this by ourselves, but we already start contacts with some collaborators which have mass spectrometry and NMR machines. For example, from Korea, we just started collaboration like a few weeks ago, and we found something interesting computationally and asked them to check their structure and their activity experimentally. So we hope to get some information maybe in January or February. So yes, there is a good suggestion, and we already started such kind of research. Thank you.
All right, final question. In the very nicely written paragraph about genome assembly, you go through the history of Sanger and and short read sequences and long read sequences and improvement. Do you think that we have a technology that produces a highly confident and nearly error-free chromosome scale or sequence, such as nanophore promises and some of the other long-read promises? But we get there, will all these algorithms be archival and useless because we'll have the whole genome sequence in one breath? And if the answer to that is yes, when is that day going to come?
Yeah, that's true. But for example, like 5 years ago, we thought that in 5 years these algorithms will be useless, but that's not happened so quickly. And I think, yeah, at some point we will be able to read a human genome in a single run. But I think at this point I will work on other algorithms because there are a lot of computational problems in bioinformatics and biology.
Good. I want to congratulate you on a nice thesis. I'll tell you the same thing I tell Pavel Pevin every time I see him. Start thinking about larger genomes than bacteria. We need you guys to scale up. Thank you.
Thank you.
As you can see, Dr. O'Brien has violated the protocol. The applicant had to take up questions. The next review will be presented by Dr. Manix Midema.
Dr. Medvedev.
Hello, can you hear me? Chair, please confirm that I am supposed to say something about the review and pose a few questions but not enter discussion with the candidate. Am I correct?
So you can announce the review and ask questions here right now.
Okay. Many thanks, dear candidate. It's a pleasure to be in your committee and to have read your excellent thesis. I've really thoroughly enjoyed reading it. I was already familiar with all of your tools, and in fact, yeah, some of them are being used in my research group. And for example, Quast is even daily bread and butter for students here at the university who need to learn how to handle next generation sequencing data. So hundreds of students here at the university are using your tool in courses, and they're all very happy with it. So in that sense, yeah, this is really a big impact. I also very much liked the combination of the genomics and the metabolomics in your thesis, as I think, well, this is a very interesting area with a lot of potential, especially now that more and more paired genomic and metabolomic data are becoming available, in which for the same samples we have both genome or metagenome sequence information and we have mass spectrometry data. And I think this will be the key not only for natural product discovery, which is very important, but also for understanding chemical interactions in microbiomes. So for example, in our own human microbiome, but also other organisms like plants, for example. So the Quast and MetaQuast tools, they have clearly made a very big impact. They include carefully chosen set of statistical parameters, and they're very user-friendly. So they're they've really filled a gap in NGS data analysis and have made a very big impact on the field. Also, the Replicator and VarQuest tools have made a big impact. I also found that especially here you are quite innovative in your approaches, your algorithmic approaches you've been employing. For example, your PNP decoy database solution to be able to also get a statistical analysis of, for example, false discovery rate. And I think also biologically, the results, for example, from VarQuest are really interesting. For example, that I think your results written in your paper about VarQuest really made very clear that there are many biosynthetic pathways that do not have just one compound as their end product, but they have a whole library of molecules as end products, and we should actually look at it that way by default. So we should always be expecting lots of different molecules from the same pathway, even if some are going to be more abundant than others. So I think this is, and we're going to see lots of those molecules in mass spectrometry data. So I think this is really an eye-opener for many people in the field. So the few questions that I would like to leave you with and things that I'm interested in to hear your opinion on when you discuss my review are basically two things. The first thing that I'm interested in to hear your opinion on is basically about the validation of the replicator and VarQuest. I think your thesis showed very convincingly that the vast majority of the predictions that the replicator and VarQuest provide are accurate, and they actually, when the replicator dereplicates a mass spectrum, it's probably going to be that molecule or something very closely related. So I think that's very convincing. What is less clear to me at the moment is basically the false negative rate. So how many compounds are there out there in all those mass spectrometry data sets, all those 100 million or more spectra, which we cannot yet dereplicate? I'm wondering, have you thought about that? Do you have ideas on what kinds of molecules cannot yet be dereplicated, and what would be necessary in the future to be able to dereplicate them? And related to that, I'm quite interested in how this can move beyond peptides to also other very relevant molecules like polyketides, which are actually quite difficult to handle with mass spectrometry because their fragmentation patterns are much less predictable. They do not have these nice amide bonds, etc. And I'm wondering whether you've thought about maybe other approaches to this. I'm thinking, for example, of things that people are working on in terms of being able to predict theoretical mass spectra, like a competitive fragmentation modeling. Maybe something like that could be relevant. I'm curious to hear your thoughts. Finally, so the second point is that I'm curious to also hearing your thoughts on the idea of matching gene clusters to molecules. I know that colleagues of you have developed approaches for this for peptides. For example, RIP quests and NRP quests. And I'm actually interested in hearing your vision about what do you think would be required to do this more generically for all sorts of natural products, and what do you think are the main challenges in this field that need to be addressed to be able to acquire real breakthroughs. With that, I would like to close and would really again like to congratulate you with your excellent PhD thesis. It was a joy to read, and I look forward to interacting with you in the future.
Thank you very much for the great review and grateful words and great questions also. I will start from the first question regarding non-peptidic compounds. Yeah, this problem is more difficult than predicting peptidic compounds due to difficult to predict fragmentation in mass spectrometer, but we recently released a new tool which called Dereplicator Plus, and it was just published in Nature Communications like 2 months ago in October. And this is exactly what I'm talking about. So it is designed for finding polyketides or even more complex compounds. So instead of looking for amide bonds, it searches for any bonds, and it has different weights for breaking different types of bonds, and so like a complex scheme of work, but it was able to enlarge the set of identified molecules from just peptidic to a lot of non-peptidic too, and we will keep on working on it. And the idea is something similar to fragmentation trees and fragmentation that you announced. So I hope this tool will be also easy to use and useful for biologists and computer scientists. And for the second question regarding matching genomic data and metabolomic data, this is also a thing which I am working on right now. We keep on improving RIPQuest and RPQuest, which were developed like 4 or 5 years ago, and of course need an improvement because they are slow and not much accurate. And we are working on different scoring schemes in them, and maybe in new approaches to estimating FDR, because controlling FDR is important for accurate results. And we are also working on new methods to combining genomic data and known molecular structures. So without any mass spec, just comparing genomic to already developed, already discovered compounds like you have in MIBIC database, but making this more high throughput. So we are working on taking a lot of genomes and a lot of compounds and finding where they're correlated to each other. So it's a promising direction, and I'm working on it too.
Excellent. Thank you very much.
Thank you.
Thank you. And Steve, are you still here? Dr. O'Brien, Steve, yes, how many time you have?
I have 25 minutes.
At least 15 minutes. You have?
Oh, yes. I will be here for another 25 minutes, then I have
okay, about that. Okay. So now, Dr. Felipe Adam, please.
Yes,
To
hello.
Dr. Felipe Adam.
Hi.
I will keep my comments rather short, and I'll hold questions for the open discussion. First, just a congratulations to Alexey on a very nice body of work. I'm particularly interested in the first half of this dissertation because it builds on some work that I did a number of years ago on assembly quality and validation. And I think Alexey said quite well in his short presentation that these prior tools had limitations that prohibited their kind of widespread use in the community. And it's very clear from the uptake of Quast, now approaching 1,000 citations, on how well of a job he did in executing his vision to overcome those limitations. In particular, I would like to highlight the impact that such a tool has, because these genome assemblies are the basis for which all of the analysis downstream happens: genome annotation, comparative genomics, even this PNP discovery. And by having 1,000 citations of his Quast tool, that indicates that there's 1,000 genomes that have now been assembled, 1,000 plus genomes that have been assembled, and the users were able to evaluate the quality and potentially improve the quality of those genomes. And so this tool has a far-reaching impact because all of those users are now generating higher quality data as a result of being able to measure the quality of their assemblies. And so I'd like to commend him for producing such a tool that's been so widely accepted and has done great things to improve the quality of genome assemblies. Overall, the dissertation was well presented, nice to read. The results were sound, it was valid, and so I was very pleased and have a positive review on the dissertation as a whole.
Thank you.
Thank you. We have one slow.
Okay. No. Я прочитала.
To the colleagues, I will comment on the Russian, in Russian on the Russian variant. Actually, I see it's sort of surprising that the candidate has to provide two texts. I would think that English would suffice, but well, probably it's somewhere in those приказы, которые были перечислены в этих самых бумагах, что диссертацию надо подавать на 2 языках. Передавайте привет вашему ректору. У меня есть некоторое количество...
I have some technical comments and editorial comments, which I have set forth in my review. The review was translated from English into Russian, and where there are some misassembles, decoy bases, and other words and phrases which do not really exist in the Russian language, and single-cell sequencing I also did not like. Speaking of technical comments, one of the section 232 mentions assembly evaluation in relation to reference strain, and E-chi strains may differ from each other. They so what are these, what types of defects are this, and how do we know these are not defects and not natural differences between strains?
So first part of the question, yes, this is only insertions and deletions, no inversions. I checked this before during the preparation to the talk. And also this was a kind of simulated experiment. So I take reads from a single stem or a single strain of E. coli and take a reference from another strain for this particular section of the dissertation, just to show that we can identify this kind of discrepancies, differences between two strains.
Okay, so you do know actually the strain, so you can estimate the error.
Yeah, so I can
based on the okay, so on your simulation. Okay. И второй вопрос это на самом.
The second question is why did you write only one thesis and not two theses? Two papers. How are the two parts of your thesis? What's the common idea for your thesis? Спасибо за этот хороший вопрос. Я согласен с вашим комментарием. Связь не столь очевидна. Может быть, мне нужно было больше сфокусироваться на единстве алгоритмов. Сравнение объекта с эталоном. Например, сборку генома с эталоном. Таким образом я бы рассматривал идентичную проблему. Спасибо, это, в общем-то, все. Также я хотел бы присоединиться к коллегам с высокой оценкой вашей диссертации. Это замечательная диссертация, которая была хорошо принята научным сообществом. И ее можно было бы представить как два независимых исследования по тому объему данных, информации, которые в вашей диссертации содержатся. But we don't have this in the paper, in the SPB SU rules.
Диссертация Гуревича.
The thesis by Alexey Gurevich, computational methods, meets the main requirements set forth by the order issued on the 1st of September 2016, order number 6821/1 on the order of awarding academic degrees at the St. Petersburg State University. Article 11 of the above-mentioned order has not been violated. Applicant Alexey Gurevich deserves to be awarded the degree of the candidate of Physical Mathematical Sciences, specialty 030109, Mathematical Biology and Bioinformatics. Thank you. The last review is mine, and it has been posted on the SPB SU website. So I will focus on some minor comments. So we are molecular biologists using this software. So our life was very hard before this software arrived. And now this our work has not been included in this reference list yet, but it will be included one day because we have been using this software already. Speaking of the Russian English texts, I read the Russian version. I understand, I know how things get lost in translation, and I feel that Alexey Gurevich has a very strong command. The entire thesis, I only found one misprint, one incorrect expression. Maybe there are some more, but considering how hard it was, the language is still clear. I had some questions, but which we have already answered here. Because the applicant is answering right away. Page 34 mentions NGA50 as the main quality metric. Does that mean that this is the most important, the most useless item of NGAX group? Page 35 shows drawing 1.7. Aligning data assembly of what does this space at the top of this drawing mean? So that's my only comment regarding translation. Mass of parent ion. Do we have this term in the Russian language? That's my question. Connected with translation. And when answering Steve O'Brien's question, Alexey already explained that one of the PNPs predicted by the program Mesothelite 1252 has already been discovered. And how is the new... So you answered this question already, so now only please answer. You only have to answer only 3 of my questions. A thesis by Alexey Gurevich, a computational method for error-prone data, metabologenomic data, meets the main requirements set forth by the order 6821 on the procedure of awarding academic degrees in St. Petersburg State University, issued on the 1st of September 2016. And applicant Alexey Gurevich deserves to be awarded the degree of the candidate of physical mathematical sciences in specialty 030109 mathematical biology and bioinformatics. Now, you should answer some questions you have not answered before. For example, the comments made by Professor Lagunin.
Remarks. One is regarding this table. She asked to add a column about NA50, but I think that he meant NG50, because NA50 is also a new metric introduced in Quast. So NG50, I added it, and you may see that there are some differences, especially in array and space assemblies, which correspond to a number of misassemblies. So by looking at the NGA50, you may see that a more correct measure of average cantic length than NG50. Also, he asked about performance of other tools. But I want to say that in case of genome evaluation, the performance is not always the main feature. Usually, the number of quality metrics computed is more important, and the performance and memory consumption running time is strictly correspond to the number of quality metrics computed. So it's difficult to compare different approaches, and I think there is no direct competitors to these tools, so it's difficult to add them to this table. Also,
Alexey, may I make one comment regarding the NGA50?
Yeah.
One thing you might consider that I've found useful in the past is to present the ratio between NGA50 and NG50, which can help illustrate how close to correct you're becoming.
Yeah, that's a good suggestion, and I think we can add it to our large list of options. Okay, the next questions. So I answered all other questions, and now let's move to Olga Igorevna questions. First one about NJ50. Yes, if you look at a single number, NJ50 is probably the best, but I prefer to look at a plot like this, and you may see that at some points blue and red lines, curves, have the same value, and this occasionally may be NJ50. But if we look at the whole figure, we may see that the blue line is always better than red one. So it's better. I personally prefer to look at all x from 0 to 100. And regarding this figure, there's a simple explanation of these spaces on the top. This is because this particular genome has a single chromosome and 3 plasmids, so we simply separate different sequences by spaces. So there is 3 plasmids on the top, and they correspond to these spaces. And yeah, and about this I answered to to Steve. Yeah.
What's about Russian term?
About Russian term for parent ions. So in English literature, previously mass spectra terminology used parent ions and daughter ions, but now they moved mostly to product ions and precursor ions. And I think in Russia this will be similar at some point, but currently I saw a lot of maternal ions and daughter ions in Russian-speaking language.
If I may add to this discussion, I've actually noticed this as well, and I've checked by Googling, and people do use this term in Russian.
Yeah.
Yeah, so it's a term. Unlike misassembly.
Yeah.
Thank you very much.
Thank you.
For the next time. Oh. So now, извините.
I'm sorry. Now, I have to read out a review by the academic advisor. Should read out. First of all, if anyone attending here not participating in the dissertation board, everyone is scared by the time limits. Any of the attendees, would anyone like to speak? General comments of the applicant. In that case, I will read out the review of the academic advisor who is not present here. I shall summarize his review. While working on his thesis, Alexey Gurevich demonstrated a strong ability to conduct research independently, was very focused, very good at using reference literature, and strong ability to interpret the results. The methodology used in his paper prove his strong skills of PC user and software developer. The main results summarized in his thesis have been published in journals included in the international, been cited many times. The results have been summarized, presented at the most important conferences in the form of papers and presentations. Professor Pevsner, since he's a scientific advisor, he's asking the dissertation board to award the degree. The review signed by Professor Pevsner, a professor of California University, San Diego, USA, senior advisor of St. Petersburg State University Lab for Mathematical Biology. Now we're going to take a 5-minute break for discussion. We'll discuss whether the board members will discuss the issue of awarding the academic degree to the applicant. And we are kindly requesting...
Can we move on?
Yeah,
please.
Sure.
I was holding a few questions. If we have time, I'll pose them to Alexey.
Sorry.
It's okay. The first question is, what are your thoughts about gene content-based validation for reference-free assembly validation? So I think of tools like Busco and CheckM for metagenome assembly. Do you have opinions on the ability of those tools to accurately estimate assembly quality? And more generally, what's a good way to evaluate reference-free assembly quality?
Actually, we embedded Busco just maybe a year ago in our tool, so we support it as an option for reference-free evaluation. I think other options are gene finding software like GeneMark, Glimmer, or something, and also mapping of raw reads to assemblies is also might give us some hints about the quality when we see inconsistent insert size or something like this in assembly. It also hints, but yeah, this problem is difficult, and I think the most accurate solutions are like Busco and CheckM. I like these tools.
Okay, thank you. And the second question on VARQuest relates to a question that was asked earlier about the false negative rate. I'm curious if there's any way to estimate the size of this search space that you're trying to explore of all possible PNPs, and if you know what fraction of that search space you're actually exploring with VARQuest.
That's a good question, difficult one. We have a tool which is currently under revision called CycloNova. It tries to de novo predict only cyclic peptides from mass spectra, and it revealed a lot of putative cyclopeptides which were not present in the database, like maybe 20 times more than we actually can find using VARQuest and dereplicator. So maybe we can use this as a raw estimate of all compounds. So we are still working about with maybe 15–20% of all compounds which we can find, but previously it was like maybe 2, 3, 5%. So VARQuest is a move forward, but it's far away from 100% completion.
Okay, thank you. That concludes my questions.
Yeah, thank you very much.
And we can go ahead, right?
Yes.
Значит, попросим выйти.
And now we are kindly requesting everyone to leave the room.
And the next point will be open vote, and that's it. If you want, any objections, it's the right time.
No objection.
No objections, right?
None here.
Not all of you. Okay. But you see, now I have to ask, and you will say yes, okay? In the next step.
Нет, ну вот я пару раз был в Нидерландах.
Yes. Can you hear?
Steve, do you hear me? Please say hi.
Hi.
Okay. So now, теперь открытое голосование.
Open voting. The broadcasting is on. Let me remind you that the decision of the dissertation board shall be considered positive If more than a half, but not less than 3 members of the dissertation board vote for it, according with Article 23 of the Procedure for Awarding the Academic Degrees at St. Petersburg State University. Appendix 1 to the order.
Mikhail Sergeyevich, vote, please. Голосуйте.
For. Yes. Да.
Okay.
I'm for.
Yes. This is Steve. I agree. Give them the degree.
Okay, great. Dr. Medema Marnix.
In favor.
Да, положительно. За.
Philippi.
Yes, in favor.
Okay, thank you very much. Я, конечно, тоже за.
And I'm also, my decision is also positive.
Глубоко уважаемые коллеги и гости, объявляю, что из 5 участвующих в заседании диссертационного совета членов совета проголосовало за 5 человек, против нет, воздержались нет. Решение о присуждении ученой степени кандидата физико-математических наук по специальности 03-01-09 математическая биология и биоинформатику Гуревичу Алексею Александровичу принято. Предоставляется слово соискателю.
Thank you very much. I would like one more time thank my co-workers and co-authors and my supervisor, and also mention my family for supporting me all this time, and of course all the members of the committee for accepting my invitation and for making this all possible, and to St. Petersburg University staff, including Larissa and Tamara, for organizing the event. Thank you very much.
Congratulations.
Thank you.
Thank you. Если не было нарушений протокола,
If no one has spotted any violations of the procedure, let me now announce this session of the Dissertation Board open, and thank you very much.